I had a question about the whole GMO concern with the sunflowers. I wondered whether there is enough of a market. So in Europe, where people are food secure, you can be a vegetarian, you can be fussy about what you choose to eat. In parts of the world where food is not so secure, is there enough of a growing market there that, for example, things like modified sunflowers would be successful?
You would think that would be the case, and yet so far the developing countries have been very conservative with their regulation of GMOs, and also the companies have been generally more reluctant to commercialize them there, to sell them there, because they want to make money, and these are the GM seed is much more expensive than conventional seed. So so far that hasn't been the case. I think it might be in the future, certainly.
Please say your name and where you're from.
My name is David Pedro from the Genome Center. This is a question for Sateesh. In terms of wheat, is there an actual marketing problem? Why the profitability? Why we're not making as much money selling wheat? Basically, there's all these gluten issues and people are not buying wheat. I just heard this morning on radio that Italians are not eating pasta as much as 20% decline in pasta because of
It's very hard to hear. Could you speak closer?
Hi, can you hear better?
Yes.
Okay. I was just wondering in terms of, is there a marketing problem for wheat? Because you're losing margins in terms of profitability that in the West people are not eating as much wheat and the farmers are not producing it. Hence, there's less demand and the market is not driving it.
Yeah, so the marketing issues are always there, like particularly for wheat. Most of the wheat that is particularly produced in Canada is exported mainly to United States, European Union, and there are many other countries. So when the wheat board was around, there was like, it was treated as like monopoly. Whether that's true or not, that's another question for debating. And there were a lot of marketing issues that were brought up. With the wheat board gone now, some of those issues probably are not existent anymore. So marketing for sure has some role in it, but it's mainly, I think, competition from other crops, like particularly in Western prairies, it's canola, where farmers get better price and yields are better. So they always choose to grow canola. So wheat is becoming more and more of a rotation crop now, that they are kind of like forced to grow it. If they have an alternative, they might choose that alternative. So.
Over here.
Hi, Andrew Reisman from Land and Food Systems. My understanding in Canadian regulation is that the PNT, Plants with Novel Traits, is the framework by which new introductions are assessed. Would a plant that was derived by CRISPR, but using an established trait in nature, even get a second look by regulators?
So that's a really good question, Andrew. Right now, Canada sort of actually has a regulatory system that makes sense, and most of the rest of the world doesn't. With that said, unfortunately, Canada is a bit player in all of this, and so the U.S. is moving more toward a Canadian regulatory framework, and under those circumstances, no, it would not be looked at. However, for most of these crops, we're talking about global crops, and they want to have satisfied the regulatory hurdles of all countries, not just in the Americas, not just in Europe. For example, if there was a gene-edited sunflower, it would have to pass muster in Canada because it's a global crop, so they wouldn't just, but they would worry about the European standards. And then they would spend $1 million or so for satisfying, if it was a novel trait, they would end up spending about $1 million to satisfy a Canadian hurdle regulatory burden. But my answer would be, I would think no.
Can I ask a follow-up? So, could Canada be a test bed? I mean, if the Canadian regulators would not stop it, isn't it a corporate decision on moving forward with its production?
So it's an interesting thing. Right now, most of the work of the companies are moving their sort of gene editing operations to the U.S. because it's an even though it is process-based. For example, the non-browning mushroom was allowed, was basically given the go-ahead without any regulation at all. It's considered to be the easiest environment, the most predictable one, and it's a bigger market. But yes, I could see certainly, I think it would be a good test case to take a disease-resistant gene or a disease-resistant mutation that occurs naturally, and move it in with CRISPR and actually see what Canada does. There's huge concerns in Europe that they're going to lose out on the CRISPR revolution and lose out on agriculture generally if their regulatory system is too strict. And so, yes, I think it would be very interesting. It could be that Canada could become a hotbed if it was, that's what happened.
Before you move to that, I want to ask a follow-on question. If genetic engineering is that much more precise and it has fewer off-target effects and maybe it's not scarring or untraceable, how is Europe going to, if they maintain their, and I think, Sally, you said, some of the first uses are going to be non-regulated uses, or what's the likelihood of that happening on agri-food? Taking a naturally occurring trait or existing.
So from my perspective, I've worked quite a bit with a number of large companies on these traits, and they really play by the rules. They're not going to do anything that would jeopardize their sort of, other than maybe Monsanto, their sort of PR and so forth. They really are careful. But I would certainly see there are developing countries that probably would see this as a way of, with good scientific expertise, I could certainly see this technology being used broadly without any acknowledgement, and it would be hard to detect. And I guess it's a brave new world.
Okay, so we'll leave that for our last panel.
Charles. This is, I guess, a bit more of a comment than a question, but I'd be curious to get some feedback from the panel. One of the groups that I participate with is made up of the ag biotech companies, and they're regulatory bodies. And one of the things we spent about most of an afternoon discussing this last meeting we had was how do you? It's one thing, regardless. Let's assume that gene-edited crops are going to be regulated in some fashion. I think it's naive to think that they're not going to be. Maybe, sure, the USDA thinks that a non-browning mushroom is okay, but if you look at Europe and other places, we're not going to. I don't think we're going to get away with no regulation. So the question then becomes, how do you measure something where you've maybe altered a couple of base pairs or made a small deletion, and when you're not even looking at it as a pure, like if you're looking at contamination within a crop and you're maybe looking for 2 seeds in 300 that have a couple of base pair changes, which is essentially the rate of regular mutation that would happen in the field anyways. How are you even going to measure this stuff?
What's the take?
Yeah, I think that's exactly the issue that Loren was alluding to, is that with precise editing, it could be a single SNP change, right? One base pair difference that in a wild population confers drought resistance or confers something. It can be edited in. And that, in terms of what we eat, I'd rather have that, honestly, than something that was generated with the same base pair change, but with hundreds of mutations through mutagenesis potentially throughout the genome. So I do think it's not an easy question, and detecting it's going to be hard, deciding about it is going to be hard, and even the language is important. I think a lot of people are using gene editing rather than genetically modified foods. to try and skirt around some of these very sticky issues. So more for this afternoon.
It will certainly be interesting to see how Canada implements its plant with novel trait, because in theory, it would not regulate some of these changes.
And just to be clear, in case other people don't know what Loren's referring to, correct me if I'm wrong, but what matters is what the product is, and if it has the same sequence as something you could get by breeding or you could get by mutagenesis, then it doesn't currently matter by regulations that you generated by CRISPR.
Question here?
Okay, I'm Sunny Yang from the UBC Pharmaceutical Science, so I'm kind of out of the field. But I was fascinated by your talk with the gene drive, Sally. So I
Closer
wanted
to your
ask-
microphone.
Yeah.
Can you move to your microphone?
Oh, yeah. So I have a question about the gene drive. So how do you choose what gene element to target? So the gene element, some of them can be very severe in terms of the fitness loss. But if you target a gene that affects fitness loss that much, then it doesn't get propagated, right, to the next generation. So how do you actually choose what gene element to select and optimize the propagation?
Right, so it's going to depend on exactly what trait you're targeting. For example, if you have a species that's resistant to an herbicide and you want to reverse that, then you could potentially edit out the resistant allele and spread that through the population by this meiotic drive advantage without really substantial costs at all. But when I talked about sterility, that one, that's a pretty major fitness cost too. So you might wonder, how's that going to work? And I think a lot of times people are talking about driving the sterility element through, if it's a recessive sterility element, so you drive the gene into high frequency, but it really only causes the sterility when it's at high frequency, so the homozygotes are produced, or other techniques so that it's dependent on the environmental conditions or dependent on something that you supply as well.
So I really hate mosquitoes, so.
Okay, mosquito.
So is it possible to initiate a gene drive and then have some kind of switch on mechanism and instantly kill all of them?
You sound tempted. So people are talking about a variety of different elements. Mostly they're worried about elements that can reverse what's gone on. So there's ways to, if you have a gene drive, but then you also have a reverse element that you're ready to use in order to reverse and counteract the change. So that's one kind of element you can engineer. Another one is an immune. So if it's going to target a site, you could spread something through that changes that site, makes it unrecognizable, but doesn't change the function. So if you want to protect some populations, you can have an immunizing drive first, and then you can have the suppressor drive. But yeah, a lot of these types of alternative possibilities are discussed in that National Academy report, including the kinds of things you're talking about, are having it driven in first and then triggered.
So one thing I think that we could use some experiments on are to see how quickly, we could do this in the lab, to see how quickly populations evolve a response to the CRISPR that basically block it. And I think it's going to be much faster and more frequent than we think. And so we have to do more clever drive mechanisms where there are two genes that have to be blocked to stop it or something. So it's sort of like the evolution of resistance, and I think that'll be quite rapid in natural populations. So a lot of these dreams might not be quite as easy as it sounds like.
Right here?
So just to reiterate that if we kill off the mosquitoes, we're also going to lose the frogs, the bats, and the birds, but keep that separate for now. My question is also about gene drive, which is fascinating. But if it's such an efficient way to move an allele into a population, why in 2 billion years of history have we not seen this before, or have we?
We have. There's lots of known meiotic drive elements or segregation distortion elements. A lot of them probably sweep through a population and we don't actually see it. So it spreads and then there's no evidence of it. But there are some that are held in check by some fitness costs. For example, a tailless allele in mice, where it spreads really rapidly when rare, but it causes, I think, male sterility at high frequency. So we do see these these alleles then remain polymorphic for a long period of time because of that counteracting cost. There's more and more evidence that there are centromere drives, that the elements that the spindle attachment process that drives chromosomes to one pole or the other are often driven to the egg or away from the egg, and that this is a very common form of drive that's revealed when you hybridize, and they have different ways of recognizing it and resisting that drive.
So just to follow up then, Loren, you said that you think it might actually be a very quick mechanism for many alleles to integrate in the population, but there's probably a counterbalancing effect.
Yeah, I think one of the things that we just don't know is, for example, in nature, we have all these natural drives, but we don't know how often they get stopped sort of very early on versus how many of them drive to fixation. And so with CRISPR-based drive, I think we really don't know at this point how often that's going to be successful versus how often they'll be blocked. And I think we can come up with ways so that it's very difficult to block them. But right now, I'm less optimistic that sort of the initial attempts, for example, to reduce disease resistance, to reduce, for example, a disease vector, that they'll actually work as planned. That, in fact, it will take round 2 before we get it right.
So, can I follow up? So, for example, if you have to target a site that's going to then be cut by Cas9, but if in the natural population there's already variation and some of those natural variants aren't cuttable, then you've got this kind of natural resistant type already segregating in the population. And if you're driving, for example, a sex ratio distorter, then you've got this natural counteracting one that will produce the rare sex. But that's where, if you are really in, we already know from the evolution of drug resistance in human drug therapy that having multi-targeted sites is the way to lessen that evolution of resistance. So I think the same would be true for these gene drive mechanisms.
So I want to switch directions slightly, but I think this is something I'm not sure of the answer now, given the talks this morning and just now. So is this really something you can do in your kitchen? So Elizabeth this morning said, it's the new PCR. And that I understand. We do that actually in high school now, I think, many times. Yet, Sateesh showed us, the gene editing platform developed at the NRC, whereas Stephen showed us a guy, I don't know if you know him, but who has a setup in his kitchen. Sally, you say it's pretty accessible, and in your risks, you raise some risks associated with the Joe Blow meddling in his kitchen. Loren, you say it's actually technically difficult. And this is going to, I think, come up in our discussion after lunch around some of the societal issues and the regulation and things like this. So starting with Sateesh, or maybe moving down, easy or not?
I would say not. And then the reason for, can you push? So, I mean, the reason for that answer is particularly our experience with wheat. When we use transgenic approach, first of all, like when we say easy, when I say on my slides easy, it is easy compared to talons, for example, or zinc finger proteins. So that's what I mean. Whereas like achieving gene targeting in an efficient manner, it's not that easy. And the reason for that is, particularly in crops like wheat, it's a huge genome. Like you saw it from Loren's slide. Like it's compared to other crop species, it's much bigger, 17 gigabase genome. And then when we were actually designing the guide RNA designer, and we were looking at for every guide RNA, what are the other off-target sites like where that guide RNA could potentially bind. And it's sometimes like tens and hundreds of places, like where there is only one base pair change or a couple of base pair changes. And we have seen even with a couple of base pair changes, these guide RNAs can go and bind and induce a double-stranded break there. So achieving like, when we say like gene editing at size, in polyploid species, it's really difficult to achieve that. Like, so we have to do a lot of work. So from that perspective, it is not easy.
Yeah, I think it depends a lot on the context, and I don't want to sound bitter about this, but if it's a student in the lab trying to do something, it never seems to be easy. To give you...
Professor.
Yeah, that's why I don't try to do things in the lab. So, if you want to do something that's been done before, like making bacteria green, that's the kind of thing that people are, in fact, doing in their kitchens. And there are a lot of sort of minor tweaks of that. And so working with bacteria is pretty straightforward. The tools are really well established. But for example, we're trying to develop a CRISPR system to use in this alga that we work on. And my students have been working simply on the design part for months already, trying to optimize that and figure out how to do it. And the level of expertise that you need to do that is probably beyond what most people are willing to develop, just in their sort of amateur efforts.
Right. So, not in my kitchen. I don't think I could do it. It's going to depend on the organism. I think we're getting the sense of it. Plants are harder. There's already gene drive proof of principle in the lab with mosquitoes and with flies. Has anybody done that with? I get the sense with flies and mosquitoes it's not as difficult as plants. The other thing is, it might not be in somebody's kitchen the way Stephen's picture showed, but there's different scales, right? So there can be a group that is really keen on developing this that has enough resources to do it. So I just think it's very difficult to know who will use it and how they'll use it and what we need to be worried about.
I think Sally's stated it well. In plants, the delivery of the CRISPR protein and RNA because you have a cell wall, so there's that delivery aspect that's challenging, as well as some of the points that Sateesh made. Some organisms, it does seem to be very facile, so I would love it if I could get one of my students to get CRISPR working in the kitchen for a sunflower.
I'm not certain what this is doing, but
Someone else has it on.
Okay, who's that? So we'll go here first and then do it.
so maybe you turn off.
If you could turn yours off for a second, if you can, and then behind you.
Okay, who wants to ask a question? Put your hand up and then we'll pick orders.
The mic behind you.
So please turn off your mic at the back. We'll go to you second. There, thank you very much. Thank you.
Okay, so my name is Dr. Scott Pownall. I've got almost 30 years of experience working in molecular biology, cell biology. I'm a geneticist by training here at UBC. I'm also, one of my hats, I'm one of the co-founders of Open Science Network, which is Vancouver's first community biolab. And I actually had, until I founded this, I did have my own lab close to my kitchen, not right in the kitchen. And one of the things that people think about, academics, they think about us versus them. And there's a lot of interest in the community about gene technologies, CRISPR, and things like that. And I've been engaging with them. And yeah, there are the hurdles there. But the other group of people that you're not also thinking about are people like me who don't have academic positions, who do have a great deal of training, who still want to continue in this technology. And it's part of the reasons why I co-founded Open Science Network, is to give me a lab that I could work in. And so there's a range of capabilities out there that isn't associated with academics as well as industry. And so, yeah, I'd like to hear your comments on that. For me, CRISPR, I've done gene targeting in mice. So CRISPR, I wish I had that back then.
Okay, Okay, Stephen?
Yeah, I think this raises a really important issue in terms of sort of the culture of innovation. If you look at more traditional technologies like cars, for example, a lot of really interesting and innovative technologies have come from people tinkering in their garage. A lot of work on electric cars, for example, came from people doing that. And so there is this potential here to be much more innovative if there's a broader population of people who are working on it. And then the question, of course, as Sally's talk made dramatically apparent, is how do we manage the risks? Because this is so new. The fact that these are self-propagating things that we're manipulating, which is not the case with cars, means it's an entirely different world in terms of the risk management.
Yeah. We've also been engaging with the Public Health Agency of Canada. So last year, I was flown to Ottawa to participate in their DIY Bio Summit, and so we had panel discussions by a number of community members across Canada, and there are more and more community labs that are opening. And I think community labs are a great way to engage the public. A lot of the people, they just want to come in and lick a bit of DNA and then they'll move on. But there are a few people that are really keen. They don't have the training, but it's not that difficult. And there's a transformation in education that's happening online, which is really allowing people to learn a lot more about biology, DNA-based technologies, and things like that. So it's there, and I think it's something that should be supported. Of course, I have the oversight. I've got the training, so I can monitor the type of work that is being done in the community lab. I can't do that in somebody's kitchen if I don't know what they're doing. So, I think the academic institutions and the governments, they really need to support community labs, just even from that perspective, but for a lot of other reasons as well.
And I just want to follow up. I think one of the things that you're pointing to is just this democratization of science and genetic technology. And it also serves as a bridge. And I think these conversations really cannot be happening in academic circles alone, because we have to be able to have well-informed discussions about what these gene editing techniques are.
Yeah, it was interesting from the academics in this room, the laughter that came up when Stephen did show that photo of the kitchen lab. I wasn't aware of DIY Bio, and yet I was buying laboratory equipment when the Canadian dollar was stronger than the US for some time. And then I discovered DIY Bio, and so I've got a pretty good biosafety level 1 lab for people to engage with, and I've been teaching synthetic biology and techniques like that. And we've been running STEM mentoring cafes at SFU and here at UBC to try to encourage women and kids, girls and kids into STEM sciences. So, there's value to the community approach.
Thank you.
Anyways.
Great comments. I hope you'll stick around for the afternoon discussion. We'll probably get into those aspects later. Other questions? Did we have any at the back? Okay, we'll go very back and then there, and then there.
Sally, one question regarding to the risk. Can you hear me?
Yes. Now.
Now it's better? Sally, one question regarding to the risk. You mentioned about controlling pests. If we tinker with this technology, whereas in comparison to what we were doing before, introducing species as control or other stuff, which one do you think is riskier? Is this a better approach? Could you expand your thoughts on this? Thank you.
So the many, many ways humans have ruined the planet in other ways, and this is another potential. Yeah, I don't really know. That's a very good question, because when we introduce non-natives to control something and then they go wild, that can have devastating ecological effects. So I do think it is important to recognize that humans have manipulated the planet in tremendous ways and many not good ways, and that it's not like it's a pristine world out there where these issues have to be evaluated in. It's a non-pristine world that we're evaluating these in. What I would like to see, though, is that there are testing done in controlled areas where you can have ensured area zones where this allele could not spread, the organism couldn't spread, so that when it gets field trialed, it's field trialed in a very controlled way, or on an island or something like that. I think, will we see it in our lifetime? I suspect so. Will we see negative consequences within our lifetime? Probably also.
Thanks. Question up here and then down in the center.
Yeah, so in CRISPR-mediated double-strand break in cleavage, what percentage of those double-strand breaks are repaired by non-homologous recombination, so that you get the effect, as opposed to non-homologous engineering or some other? Because creation of double-strand breaks in the cell is mutagenic. It's mutagenic genome-wide, and so given the efficiency of those two competing processes and everything else, every other way that you can repair a double-strand break, I mean that's a big problem with the efficiency of Cas9. Are these going to be wildly mutagenic?
Elsewhere in the genome?
Well, it's not necessarily elsewhere in the genome, because it's going to, if it just targets the homologous segment, and you've done a good job of making sure that that's
right,
true.
No, but I mean that not, but like bacteria induces
yes.
SOS and mutagenic polymerases, and in eukaryotic cells it's Rad52 and that pathway and old gamma and very low fidelity polymerases that are sort of last ditch repair pathways that sort of, and they're mutagenic. And so that's, is that considered?
So just when it targets the homolog and it does that correctly and induces a double-strand break, you're absolutely right, but there are other neighbor end joining is a very common way of closing that gap. So it depends on exactly where that is. So the efficiency of homologous repair versus not definitely depends on what's targeted, and it has been shown to be as efficient as 98% through homologous recombination repair, and down to close to 50%, which is much less spreadable. But the other thing but this is also where one has to think about where one's targeting. If one's targeting an essential gene, then an indel, which is typically created by neighbor end joining, could be lethal. And so in that case, then it's more likely that the recombinational repair pathway will be used. But you're absolutely right, and so it depends on where this element is targeted. It depends on what that gene does and whether or not indels will be tolerated in that gene or not, whether or not you'd see the actual spread of the gene element or just blunt end joining.
And if I can just follow up on that, there are sort of two scenarios in which one might use CRISPR, how it's used. In one, you're just cutting someplace and allowing the cell's natural repair mechanisms to fix that by the mechanisms that you're talking about. In the other, you actually introduce a repair template that's homologous to the neighboring regions, and that directs the repair. And of course, that can dramatically shift the efficiency of homologous recombination relative to other pathways.
So it can be shifted to near 100% with that added template at achievable levels?
Yeah, that's right.
It still must be wildly locus dependent.
Yes.
Yeah, absolutely, and organism dependent too.
And even the time in the life cycle depends.
Oh, yeah.
Okay, so we have a question down here. And then right there.
So just switching the focus a bit. So I know a lot of these genomes that we're working with, the genomes haven't been fully sequenced. Do you know of anywhere where this technology is being used to help complete those genomes?
I don't.
You win the prize for the quickest question. Okay.
Hello, I'm Christian Buchwalder from UBC Pharmaceutical Sciences. As a chemist, I'm a bit out of the field, but I'm very interested in this whole topic. I was having an idea of, like, can this be misused by a small group or an individual in a kind of act of bioterrorism or so? But then, especially with the gene drive, if the group wanted to, say, eradicate certain species or certain ethnic group or so, can we, however, use this technology to revert this process also? And then if, say, government groups have the resources to then just undo this whole attempt?
Well, so in theory, there are ways to generate reverse drive elements, whether it would be fast enough to undo. The other thing is to not go too fearful, as Loren says, natural resistance may well evolve. The other point is that in organisms that reproduce very slowly, we're going to have lots of time. This is not an element that we've got to worry about converting humans rapidly. We'll notice it. So it's really fast generation time, things like insects, where we may not have much time to act before the effects are seen.
Any other questions? So I have a question. So I'm fascinated by the idea of using gene editing to bring back, species that have become extinct or threatened, things like this. Do you ever think that we'll see a World Wildlife Fund or Environment Canada stepping up and leading that work and
I hope not, because that's enormously expensive relative to the things that are cheaper that we're not doing right now, which is really conserving more, using land less, protecting more species at risk. So rewilding is definitely intellectually interesting, but it's not a conservation approach.
Oh, I want to see a mammoth. In my lifetime.
I want to see one too.
I don't think
that
that
might change the food
necessarily
chain.
governments should be doing this, but I certainly can't imagine that there won't be private funders who, like to do things like, go to Mars that they wouldn't want to see. One of these more recently extinct organisms, like a saber-toothed tiger or a mammoth or so forth, where we can sequence the genomes and actually have a good idea of what the major gene changes are. I think that's possible, and I wouldn't want to commit taxpayer money to it, but I would be happy to commit someone else's money to it. So.
And we do it on an island, right? Jurassic Island.
Vancouver Island.
Other questions? We've got a few more minutes. 2 minutes. Well, I've got another question. What's yours?
I was just wondering if wheat growth in Canada is expected to do worse with global warming, and if the heat tolerance genes are known and could be targeted, I guess similar to the sunflower.
I think that's one of the worries. Like the acreage is going down every year, and then some of the farmers. We generally do visits like in summer to a lot of farmers, and when we speak to them, there is one is like the FHB menace is becoming really big, and then that is actually driving farmers off growing wheat. So and also the productivity and profitability. So those are the things. When we talk to them, maybe next year I don't want to grow wheat. We hear that quite often. So. I think from that angle then improving the productivity and profitability for Canadian farmers is really important, which will encourage them to grow wheat again, I guess. And then this is not just in Canada, this is actually worldwide. And then this concern was, I think, noted by the G20 countries. And then a couple of years ago they came up with this wheat initiative, where the whole goal of wheat initiative is to do innovation or encourage innovation where wheat productivity can be increased.
One comment I just wanted to add this is that the arable land is increasing in Canada due to climate change. One thing though is that high value crops from the US are moving into Canada more and more, and so what we're seeing is that of course canola does very well, but it's being pushed north by the maize soybean cycle. So where wheat goes in this, I think, it's how it competes with other crops, as Sateesh has mentioned, but there will be more land overall.
So I've had the time's up sign, so that I think is a good way to, a positive note to end on. I think I see it as positive. So I'd like to thank the speakers one more time. Thanks very much, and thank you for your great questions.